Hello again. It's me. I know it's been a while, and I'm sure you've missed me in my PowerPoint presentations. So yesterday, I had you look at and read the chapter, and now I was just going to go through some of the different stuff about bacteria. You may have noticed that I've upgraded. I'm a little fancier now with my. I've upgraded from PowerPoint 2002 now to probably whatever the newest version is. So I'm sure there'll still be some technical difficulties, as is the case. But anyway, bacteria. And I just want to review bacteria, which we went over a week or so ago with our chart. Remember, they are prokaryotic, unicellular, and they could be heterotrophic or autotrophs. Heterotroph, meaning they have to obtain their food from outside themselves. Autotroph, where they're making their own food as an energy source internally. We'll get to some other details of those later. And the other aspect of bacteria, these prokaryotes, difference I wanted to highlight is that they have circular DNA. Eukaryotes have linear DNA in that classic kind of double helical shape. Bacteria have a circular genome, just kind of circular. So again, prokaryotic, unicellular, heterotroph, or autotroph, circular DNA. All right, the cellular features of bacteria. If we start on the outside, bacteria can have these little hair-like structures, pili, that are going to aid in smaller movements through these liquidish mediums. And they're going to have some, but not all, will have a flagellum, which is this tail-like thing. And flagellum are kind of cool. If you know the movement, actually, it's kind of a circular movement at the base down here. And as the flagellum moves, it rotates the flagellum, but it kind of gives it a corkscrew mechanism through the fluid, which helps bacteria move through things that are not quite as fluid, that are a little thicker than your normal fluid, like water. Yeah, that's kind of cool. They have this little circular movement. And then bacteria are going to have cell membrane, as all cells have a cell membrane. Some will have a cell wall, which is important in antibiotics. Cell wall. It's one major difference that we don't have that they do. Some of them, but no. And then their DNA, again, they're prokaryotes, so they don't have any membrane-bound organelles. They do have the DNA, and it's contained in this region called a nucleoid. And you can see here it's all messed up and bundled together. But if you were to get it out and stretch it out, it would be a circular motion. And then the one organelle they do have, the ribosome. It's one similarities that they have. All the processes they do within the cell, I mentioned this way back in the beginning of the year, they're going to be able to do with enzymes and proteins they have within the cell. Some of them are going to be embedded on the cell wall or the cell membrane. They're not using any of these organelles, the mitochondria, endoplasmic reticulum. They can do everything they need just within this cell. So even though they're basic, they can do anything that we can pretty much. Just not quite as efficient and not as good, I suppose. All right, bacteria, when we look at the classification, are going to be classified into three different types based on their shape. The first one, the round shape, the cocci here. Sometimes they'll join together and get these long kind of linear structures going. Second one are the bacilli. These are these rod-shaped bacteria. See these under a microscope. Pretty cool looking ones. And then the spirochetes, which are probably the nastiest ones. All three of these, by the way, can cause disease in humans if they're in the wrong spot. But the spirochetes, as far as I remember, are the ones they kind of they can do the nastiest ones. Some of the nastier diseases. I think cholera, I believe, is spirochete, which I'll get to in a different talk. do want to say that bacteria, where you can find them anywhere, right? They're covering us on surfaces all over the place. All right, so the oxygen requirements, some new terms, because we can have anaerobic or aerobic bacteria and some combinations of it. There are fancy terms here. You have obligate aerobes. This comes from the term obligation, which you need to do it, or it's a requirement. And then aerobic, remember, is those that need oxygen. So an obligate aerobe, that's going to go through cellular respiration. It needs oxygen in order to survive. Without oxygen, it's going to die. It's going to essentially suffocate and die. No respiration, no ATP equals death. Then we have the obligate anaerobes, and they are, again, obligation requirement. And these do not need oxygen. So oxygen will actually kill these. They're going to go through fermentation. Tetanus is a bacteria that is an obligate anaerobe. And we'll get to more details about tetanus a different day, but it's oxygen will kill it. So hydrogen peroxide is nice in killing that. Helps oxygenate wounds. But some will die in oxygen. You don't usually think about oxygen killing things, but it does. Then facultative anaerobes are ones that can go either way. By default, they're going to use oxygen cellular respiration for ATP. But if the conditions are necessary, they can go into and use an oxygenless environment. Then we have our different types of energy source, which is a difference for bacteria. We have our heterotrophs, which are normal, again, getting energy just outside themselves. We have our photoheterotrophs, which are going to use a combination of autotrophic. They can use light as their energy source, or if needed to, they can use some sort of other energy source outside themselves. So it's kind of a combination of both, which would be kind of neat. If we could humans could go between the two, it'd be kind of fun. We have our chemoautotrophs, which are going to use, they're chemical feeders. They're going to use some sort of energy from chemical. Usually they're in the volcanic vents in the deep ocean. There's no light, and organisms are scarce, but there's plenty of stuff like some sulfur-containing compounds spewing out of the ocean. And then you have your classic photoautotroph, which is just going to use light as their energy source. I think that's it. Nope, sorry, one more. Bacterial uses as energy source. And then we can use the bacteria. They're not always bad. I think I mentioned earlier that bacteria pretty much covering every surface. It's covering your body from head to foot. It's everywhere. There's no way we can't avoid bacteria. If you keep bacteria in the right spot, it won't hurt you, won't harm you. So when bacteria gets in the wrong spot, or you get one of these particularly infectious bacteria, that can cause problems. But there are many different uses for bacteria. Your book highlights nitrogen fixation and decomposition. Bacteria can take inorganic material or nitrogen N2 and fix it into compounds so that other animals and plants can use it. So otherwise, nitrogen, if we don't have nitrogen, not good for us. And it can help recycle dead material back into the soil so that other things can use it for energy if needed. Plants, some animals, whatnot. Really, ultimately, plants and then animals. We use it for food. This picture didn't come out very well. But you have yogurt right on this side. Use it for cheese. Any kind of fermented food, sauerkraut, if you like your Reubens. And wines and whatnot. We use bacteria for that. So a lot of products, maybe even in your fridge, used for bacteria. And a use I like to highlight is we can use bacteria for medicines and some different things. Insulin is now produced using bacteria. Back in earlier days, the 80s, they had to harvest it from the pigs and pig pancreases, and that was a lot more expensive, and I don't know that it was I don't know how much they would harvest from per pig, but they did. And now they grow bacteria, and bacteria will release it as a waste product. And then you get your insulin, and it's more effective, more efficient, and I believe it's got to be a little bit cheaper. And we'll talk about how they change or manipulate bacteria to make what it wants. But it's not the only thing that bacteria now are used to do. They're helpful in some vaccines. They're helpful in just making enzymes that scientists and industrial people will use to make other things. Anyway, so they're not all bad. They can be very bad, but it's that's that 1% of bacteria that gives all the rest the bad name. Most bacteria is good. They're like your little buddy, your little friend, as long as you treat them nicely. Anyway, that's a wrap. Don't forget to like, to subscribe, and to, I don't know, enjoy it, I suppose. See you.